Hello, so this is the last section of chapter 5, enzymes. So, although biological molecules possess much potential energy, it is not released spontaneously. So, we have this thing called an energy barrier or activation energy. And so, we can see here, if we can get that hill downhill, it is a downhill reaction. It would be an exergonic reaction, but we got to get it over the hill first. And that hill is referred to as the activation energy. So we can think of the activation energy again as that energy that we need to make that molecule unstable. It's kind of like sitting. If you were to sit on the top of a hill on your bike, you're not going to just go down the hill. You got to push off, right? That little push to get you over the crest. And once you're over the crest, then you're good. But you've got to put that extra energy in. One way that we can speed up the reaction in the chemical world is to add heat. That heat increases kinetic energy, that causes more of this agitation, which allows the chemical bonds to break easier. The problem in the biological world is when you add heat, it kills the cell, and it kills the cell because it starts to unravel or denature proteins. And without proteins, we cannot maintain our structure. We cannot maintain homeostasis. We cannot get chemical reactions to occur fast enough to support life. So things go all sideways. So here is an example of that activation energy. So you can see that hill that we've got to get up and over that hill. So our reactants are higher in energy than our products, but we've got to get up and over that hill first. Now, what enzymes do is enzymes are considered a biological catalyst. A catalyst in chemistry would be an element like calcium, or actually, calcium is not really one, but well, I guess you could say it is. There are some things with calcium, but I was thinking more like zinc or copper. They help things to speed up. In biology, we tend to use enzymes. All right, so enzymes are like 98 to 99% of our enzymes are proteins. The other couple percent are RNA molecules. So here is a side-by-side comparison, and you can see that our activation hill here, or our hurdle to get up and over, is much larger than with our enzyme. So this speeds up the rate of our chemical reaction by lowering the activation energy required to get from reactant to product. Here's another way of describing that particular reaction. So we have a graph here, and you can see the black line that activation energy is from here to here versus, and so that would be without your enzyme. You can see here B is your activation energy with an enzyme. Okay. Again, our energy of our reactants or products didn't change. It's still exergonic. It's just the amount of energy we need to put into that reaction is a lot lower with the enzyme. An enzyme is very selective in the reaction it catalyzes or speeds up. It has a specific shape that determines its specificity. The specific reactant that is what the enzyme acts upon is called the substrate. The substrate fits into the region of the enzyme called the active site. Enzymes are specific because their active sites fit only one specific substrate. We always tend to use this analogy of a lock and key. You have many keys, but it doesn't mean you can open every lock. Each specific key will open a specific lock because of its fit, and that's how enzymes work. They have an active site. It has a certain shape associated with it, and only the molecules that will fit into that by that pocket it forms will then be acted upon. So here is that active site I was talking about. Here is our enzyme. We tend to name enzymes with the ending ASE. So this is sucrase. And then we name the root, you could say, or the prefix of the word on the substrate. So in this case, our substrate would be sucrose. Sucrose is a disaccharide. It will fit into this enzyme called sucrase. It will cause the enzyme to change shape and put stress on that covalent bond. We will need water. This is a hydrolysis reaction because we're going to break a chemical bond, which means we got to put, I always talk about our end caps on there, or we have not met octet and we won't be stable. And then we have broken down our disaccharide into two monosaccharides, glucose and fructose. Lactase works the same way. So lactose is a milk sugar you may have heard of. We're going to talk a little bit about that. So lactose is a disaccharide. We have an enzyme called lactase, and it breaks that disaccharide into monosaccharides. Same basic idea here. So for every enzyme, there are optimal conditions that affect the enzyme, and we want the enzyme to be most effective. We generally talk about enzymes are most effective in their native environment. That is the environment you would normally find them in. Temperature does affect molecular motion. Again, it's going to increase the kinetic energy. But again, once you get outside that enzyme's normal temperature, it will start to denature. So for our enzymes work best between 35 and 40 degrees Celsius, which is body temperature. The optimal pH for many enzymes is near neutrality, which is 7, but there are some enzymes, for instance, the ones found in your stomach, will only work in a very acidic environment. Once you get them out of that acidic environment, they start to denature and will not work anymore. So this is kind of a chart that I put some graphs in. We're going to be doing a lot of graphing here in this portion of the class. And so we have this graph here, and you can see the enzyme activity. It peaks out right here. So we could say that our peak temperature is, right there between 30 and 45. If I want to go all the way, I wouldn't even go 45. I would go more like between about 43. pH, so pH peaks out, we'll put it right here. So probably close to neutral. And then we have concentration. So concentration, enzymes will work faster and faster and faster as we increase their concentration, whether you increase the enzyme concentration or the substrate concentration. But again, enzymes can only work so fast. So once you have saturated, that means you have as much substrate as you can put in there. We can only have one molecule bound to an enzyme at a time. So once all the enzymes are full, we can't go any faster. So that would be saturation. Many enzymes require a non-protein helper called a cofactor, which binds to the active site, and it functions as a catalyst. So in this case, we have this non-protein coenzyme that's going to kind of go into that binding site to help the pocket have the correct fit with that particular substrate. Some cofactors are zinc, iron, or copper. If a cofactor is an organic molecule, meaning it has carbon and hydrogen, we call it a coenzyme. And this is what your vitamins are. Your vitamins, many of your vitamins are coenzymes. We'll talk about some of these vitamins when we get to cellular respiration. A chemical that interferes with an enzyme activity is called an inhibitor. So it's inhibiting, it's preventing it from doing its job. We have this term called competitive inhibitors, and they are molecules that kind of fit into that binding pocket. And so they get in there and block the actual substrate from working. It would be like having a key that kind of fits into the keyhole, but it won't turn the lock. And you can't put the correct key in the hole while the bad one is in there. So it lowers the productivity of that enzyme. So here we can see we have our substrate binding into our binding pocket. Everything works good. We break it apart. Here we have an inhibitor, and notice that it has that shape there, but it's not the correct substrate. And so when it's bound, the correct substrate can't get in there. So again, they're going to go in and out. They don't just sit there and permanently bind. There are some chemicals that do that. For instance, carbon monoxide will permanently bind hemoglobin, which is why carbon monoxide is so bad. Right, but for most part, they come in, they come out, they come in, they come out. Right, so but while they're bound, we can't bind with the correct, we cannot bind with the correct substrate. So an example of competitive inhibition. So with our competitive inhibition, I have an example. I have a lot of students that are like, can I have an example of this? And with that example, we have an enzyme here, and normally we would bind to succinate. And with succinate, you can see the shape kind of fits into this particular enzyme, and then it forms fumarate. Malonate looks very similar, has a very similar shape. And so it binds, and as it's bound, we don't get a product. So it is competitively inhibiting this enzyme from working properly. Non-competitive inhibitors bind to the enzyme somewhere other than the active site. They change the shape of the active site and prevent the substrate from binding. So I always like pictures. So here's a better look at this. So here is our normal, things are going well, right? We have our active site, we have our substrate, we bind. Here we have our competitor, totally blocks our substrate, not getting in. A non-competitor binds to a different site in the enzyme. And what happens is when it does that, this site here will change shape. And then your substrate will no longer fit. So it's causing a shape change in your enzyme. So since it's not competing directly for that binding site, we call it a non-competitive inhibitor. It's still slowing down the rate at which that enzyme is working. So enzyme inhibitors are really important in regulating cell metabolism. In some reactions, the product may act as an inhibitor of one of the enzymes in a pathway that produce it. This is called feedback inhibition. And so if we look here, we have a lot of this yellow substrate. Again, so the odds of it being bound to the enzyme increase, right? The more you have, the more collisions you have, the more likely you're going to have a chemical reaction. Okay. Now we have our product. It makes orange pies. Now, as we continue to convert our yellow balls to orange pies, eventually we have too many orange pies. And so we have a secondary binding site on that enzyme that when this concentration goes up, the odds of it being bound to that site increase as well. And so now it's saying, we do not need any more orange pies. And so again, that active site changes shape, and now we're not going to continue to convert the orange pies. Now, eventually the orange pies will generally go into a different chemical reaction. And so those numbers will come down, and then the odds of this being bound go down. And then we say, oh, we need more orange pies again. It's a lot like your thermostat, right? Your thermostat doesn't like keep your house within a certain temperature by adjusting the air conditioner and the furnace. It's one or the other. So let's say right now we're using our heat. The thermostat detects when you've reached that maximum temperature, and it shuts off the furnace. And then your house temperature decreases again, and then it kicks on the thermostat again, and it keeps bringing it up. And this is kind of how this type of regulation is occurring. Here's another example of this. So we have a series of chemical reactions. The reactant makes the product is now the reactant to the next chemical reaction. We have enzymes at each step in order to speed up the rate of these chemical reactions. Now we're saying, well, if we have too much of D will actually tell enzyme 1, we don't need any more. You don't need to keep going from A, B, C to get to D because we have enough D. So let's stop this train right here. This is referred to as negative feedback because it is negatively affecting those chemical reactions. It's stopping it. Again, as D gets used up into another chemical reaction, then it will decrease the probability that it is bound to enzyme 1, and then we will move forward again. So I hope this was a better explanation. I know this topic tends to be a little bit harder on feedback inhibition. I hope you understand enzymes a little bit better. We are doing a lab on enzymes. Hopefully you watched this presentation before you did the lab because it would be helpful. If not, I can't really say go back and watch it because you wouldn't get that message now, would you? So if you have any questions about enzymes, again, I have some supplementary videos for you to watch. But either way, if you do have questions, do send me an email or we can schedule a Zoom. Thanks.